Good morning, children. In the last class, actually, we were started the discussion with respect to the competitive part. Okay, so but the thing is, this chapter is a very big chapter, as well as when it comes to the weightage of the theory, even including competitive also, minimum 3 to 5 questions. Sometimes they were given up to 6 to 7 questions also. So that's why we will discuss a few more questions in this class. Okay, yeah. So in this one, first question: which statement is incorrect about transcription? Okay, so which statement is incorrect? Underline the term incorrect. Okay, so that means whatever they have given the 4 statements, out of these 4 statements, the 3 statements are correct and only one statement is incorrect. So we should identify that one. So let me to write those questions, the options which is given for this question. The first one, A. It is the process of copying genetic information from one strand of DNA into the RNA. It's true or it's correct. Okay, then option B, adenine pairs with uracil instead of thymine. Again, it's correct. Okay, then third option, an insertion factor present in RNA polymerase initiates transcription. So option C is also a correct one. Option D, it takes place in 3' 5' direction. It is incorrect, because so generally the transcription takes place from 5', 3' direction. Okay, but they have given here 3', 5'. So that's why option D is the right option, but it is an incorrect statement about the transcription. So that means this question is related to the topic transcription, or we can also say the logic distractor, the transcription. Okay, yeah. The next question, the second one. Okay, the second question, so this is also related to the transcription. So let me to write the question. The DNA template sequence of CT, GA, TAA, GC is transcribed over messenger RNA is. See, I told you in the earlier classes also. Okay, so whenever it is a DNA, then it will be ATGC, but a transcription, after the transcription, so the messenger RNA, that RNA containing the nitrogen base is AUGC. So wherever the CGA present, there will be no changes, but instead of wherever the present, the thymine, there we should be generally identify the uracil. Okay, yeah, so let's check it out. Options with respect to this one. So C to the G and T to the A, and G to the C and A to the U. Okay, then again here what they have given is T, A, and in the DNA I'm telling about CT, GA, TAA, GC. So when it is after the converted into messenger RNA, instead of the opposite, wherever it will be present, instead of T, all it should be replaced with the U. So that's it. Okay, so opposite you should identify. Okay, so then which is the right option? G, A, C, U, A, U, U, C, G. So that means option B is the right option. Okay, because here the messenger RNA, okay, already transcription is completed, that's why transcribed. Okay, the next question. So third one, so this is also related to the transcription only. Reverse transcriptase. See, while we are discussing about the transcription, I told no. So there are some viruses like HIV. We can see the reverse transcription. Okay, so reverse transcription, it is because of the presence of the enzyme reverse transcriptase. So that what is the function of this one? Reverse transcriptase is RNA dependent DNA polymerase. Because I told no, generally during molecular, the central dogma, the DNA undergo replication and it will be gives rise to the messenger RNA, that is a transcription. Then later messenger RNA will be gives rise to the particular protein, that is a translation, transcription and translation. But what about in case of the the HIV like that, due to the presence of reverse transcriptase, so immediately after the DNA is get converted to messenger RNA, but immediately it will be undergo, so that is again the DNA. So that means the RNA dependent DNA polymerase. Did you got it? Yeah, this is the right option. Option D is the right option. Okay. The next question, a functional unit of gene that specifies a synthesis of one polypeptide is. See, while I'm telling about the units of the genes, I told no what is the meaning of the codon, recon, cistron, muton, like that. Okay, so when it comes to the functional unit of the gene that specifies the synthesis of one polypeptide is nothing but cistron. Okay, and you know the meaning of the codon. Okay, triplets, that is a combination of the nitrogen bases in a 3, that is generally ATG, TUC, like that, whatever it may be. Then the recon, it's a function unit of the replicon. Okay, then muton is responsible for the mutation. But here the cistron, a functional unit of gene that specifies a synthesis of one polypeptide, is called cistron. Okay. The next question, it is also related to the transcription only. So, which is incorrect about the monosystonic structural genes? See, while we are discussing about the transcription in prokaryotes and transcription in eukaryotes, I told no. So, monosystonic genes and polysystonic genes, you must have heard about. Okay, and you studied already in the class also. I hope you can remember. So here the question is, which is incorrect? Then you should identify the incorrect, underline the term incorrect about monosystonic structural genes. Okay, let me to read the options. It is seen in prokaryotes. Okay, so first option A is only, it is incorrect because monosystonic structural genes are found in eukaryotes. Polysystonic genes are found in case of the prokaryotes. Okay, and remaining and all correct. It is a type of structural gene. Yes, it's correct. It is also known as the split genes. Yes, it's correct. And the coding sequences are interpreted by introns. Yes, it's correct. Which is incorrect? The first option A is incorrect about the monosystonic structural genes. Okay. So this is exactly the logic distractor of the topic. Yes, that is nothing but the transcription in prokaryotes as well as in the transcription in eukaryotes. The next question. So just now I told no the terminologies, meanings. The portion of a eukaryotic gene which is transcribed but not translated is called introns. Okay, the portion of eukaryotic gene which is not transcribed. Okay, so whenever it will be not transcribed but not translated, so then it will be introns. Okay, so direct question, this is actually, this can be asked for 1 marks question also, no need of confusion. The next question, the 7th one, spliceosomes, the complex molecule necessary for processing of messenger RNA, are not found in cells of, so this is nothing but the prokaryotic cells. So this is also related to the transcription in prokaryotes. The very best example, that bacteria. Okay, so are you getting what is this exactly question? The spliceosomes the complex molecule necessary for processing of messenger RNA are not found in cells of bacteria, because bacteria comes under the prokaryotes, but spliceosomes can be identified in case of the eukaryotes. Did you get it? Yeah. The next question, the 8th one. Okay, so this is also related to the transcription only. The direct one, the direct question. Methyl guanosine triphosphate is associated with. See, while we are discussing about the post-transcription, okay, the post-transcriptional process, I told no, the capping and tiling, you must have heard about. Okay, so in the while we are discussing about the post-transcriptional process, so that means the methane guanosine triphosphate is associated with, that is MGT is associated with capping. Okay, capping. The remaining and all, there is no need to confusion. You know what is the meaning of the point mutation. The mutation is happens with respect to the particular sequence and tautomerism. So you know what is the meaning of tautomerism. Generally it is look like a the repeated sequences. So in this chapter we were not discussed about the tautomerism anyway, and Okazaki fragments when it comes to the unwinding nature. Okay, that is a during transcription 4, isn't it? Then the tiling. Okay, so which is the right option? Option C is the right option. The next question. So this question is related to the genetic code and translation. Till now whatever we were discussed, it's about the transcription. Then after, so this question is related to the translation. Initiation codons for protein synthesis are, okay, so I told you in the beginning also, when it comes to the initiation codon, either it should be AUG or GUG. Okay, so this AUG and GUG is commonly called initiation codons. Okay, well, we are discussing about the characteristics of the genetic code, I told no. So I think you have written the in your notes also, there's initiation codons AUG and GUG. So here they have given beginningly GUG and AUG, no problem. The next question, genetic code and the translation, it is also related to that one. So genetic code is said to be degenerate because, see, this question is direct one actually. So just now I told no the characteristic features of the genetic code. Okay, so genetic code is said to be degenerate because one amino acid is coded by more than one codon. Okay, if you see that table, whatever we were given the the combination of the codons which is responsible for the synthesis of amino acids, I told no there are 64 in the codons. Out of that one, 3 are nonsense and 61 codons which is codes for the 20 amino acids. Okay, so but here when it comes to the degenerate, the characteristic, one amino acid is coded by more than one codon. So that's why it is also known the genetic code is said to be degenerate. Okay, which is the option, right option? Option B is the right option. The next question, termination. Okay, the termination of polypeptide chain is brought about by, while we are discussing about the codons, just now I told no, out of 64 codons, 3 are nonsense codons. They are also known as terminatory codons. Okay, so that means which is stops after the particular protein synthesis, that process should be stopped normally. So then there are 3 terminatory codons or the nonsense codons which is related to the stopping of the process, that is UAA, UAG and UGA. Okay, next question. So this question is related to the match the following actually. So again here the genetic code, that is a codons, with respect to the particular amino acids. Okay, the match the codons with their respective amino acids and choose the correct answer. Here only they have given UUU, it is actually phenylalanine. Okay, if you see the table, okay, there are 64 codons table is there, no, in your textbook or in the notes. If you see that one, just once, okay, then you can come to know. And GGG, it is nothing responsible for the proline. And UCU, it is a serine, related to the serine. And the CCC is nothing but the again, okay, so that is nothing but the proline. Now the AUG is nothing but methanoine. So now match the correct one, the match the codons with their respective amino acids and choose the correct answer. Option A is the correct answer. Okay, so with respect to the, that is a codons, triplet codons with respect to synthesis of amino acids, or they are responsible for the synthesis of particular amino acids. Okay, next question. So this is related to the genetic code and translation. Direct question, actually it is a messenger RNA, that is mRNA is polymer of ribonucleotides. So that is nothing but, I told now, in case of the DNA, deoxyribo, okay, deoxyribo sugar will be present. What about in case of the RNA, ribo sugar? So then when it comes to the messenger RNA is a polymer of ribonucleotides. Okay, it is actually a direct question. The next one, so this question is related to the genetic code and the translation. So this is a purely related to the, the discovery of the genetic codons. Dr. Trna and his colleagues synthesized an RNA molecule with repeating sequences of UGN bases, that is nothing but UG, uracil and guanine combination actually. Okay, it produced a tetra, that is nothing but tetrapeptide with alternating sequence of cysteine and valine. It proves that codons for cysteine and valine, they are nothing but UGU and GUG. Okay, because just now I told no, so in this UGN bases, uracil always combines with guanine. Okay, and so guanine always combines with the uracil. Okay, so then which is the right option? That is nothing but option B, UGU and GUG. So next question, so this is also related to the genetic code and the translation. Which of the character is not applicable for transfer RNA? Okay, see here they were asked, not applicable. Out of these 5 statements, only one is not applicable and remaining and all applicable. Okay, so let me to read one by one. Option A, it is the smallest of the RNAs, it's correct, it is applicable. It acts as an adapter for amino acid. There is a tRNA, is acts as a adapter molecule, right? Yes, it is applicable. And it has a cloverleaf-like structure, H, it is applicable, it's correct. It bears NODOC, yes, it is also applicable. It is the largest of RNA, no, it is the smallest of RNAs. Okay, so that's why option E is the right option with not applicable statement with respect to the tRNA. Okay, the next question, which is not involved in protein synthesis? Okay, see I told now, so this is a common sense as well as related to the central dogma of molecular biology. DNA undergo replication, will be gives rise to the messenger RNA, is known as a transcription. Okay, then after messenger RNA will be gives rise to the undergo, that is a translation, then it will be gives to the protein synthesis. which is not involved in protein synthesis means, so don't think that here it is, I mean the how it will be, because while we are finding the answer we should identify most appropriate one. Okay, so that's why the transcription, okay, between DNA and RNA, after RNA will be transcribes. Okay, it is a transcription and translations. Okay, after the translation the protein synthesis will be takes place, but here what they are asking, which is not involved. Okay, so that's why option A is the right answer. Okay, because during protein synthesis, initiation, elongation, termination, surely it will be takes place, isn't it? But after the messenger RNA, it will be gives rise to the, I mean the particular protein. The next question, choose the wrong statements related to the protein synthesis. Here it is also, see whatever they have given the 5 statement, among these only one statement is a wrong one and the remaining are all correct. After that is uncoiling of DNA molecule, one strand acts as a template for the formation of messenger RNA. Yes, it's true. Okay, so then B, in the presence of DNA polymerase enzyme, the messenger RNA is formed based on the triplet codes. Yes, but see, this is the right option, the wrong statement. Okay, because in presence of DNA polymerase enzyme, messenger RNA is not formed based on triplet codon, isn't it? Yeah, so that's why. So how it will be possible? Both will be, isn't it? So that's why option B is the wrong statement related to the protein synthesis. Okay, the remaining statements are it's correct only, because the messenger RNA that leaves the nucleus reaches the cytoplasm and gets attached with the 30S ribosomal unit. The amino acids are transferred from intracellular amino acid pool to the active ribosomes by the tRNA. Translation is the process in which proteins are synthesized from the RNA. Yes, it's correct. Okay, so that means out of this 5 statement, option B is the wrong statement related to the protein synthesis. Remaining and all true statements related to the protein synthesis. The next question, see here, you should identify. So when we can identify this process, the reaction, so this is related to the translation. Okay, the first process during the translation, amino acid plus ATP, so amino acyl AMP plus P, that is a depict. So this process is also known as amino acetylation, isn't it? So amino acetylation, it is also known as amino acid activation. Okay, so it is a direct question actually, no need of confusion. Next question, so it is also related to the genetic code and the translation. What would be the anticodon on the tRNA? RNA means that is AUGC, that picks up and brings the amino acid methanoin. That is nothing but, just now I told no, anticodon you should identify. Okay, so generally whatever it will code for the amino acid methanoin, AUG, isn't it? But for that they were asking the anticodon, so then it is a UAC. Okay, the next question, it is also related to the genetic code and the translation. UTRs, untranscribed regions. Okay, where it is present generally, untranscribed regions are present in messenger RNA. Okay, it's a direct question again. Fine. After the transcription and so generally messenger RNA will forms, and messenger RNAs get converted to a particular protein, that is nothing but translation. But after the translation also there are subunits will be present. So where we can find and transcribed regions are present in messenger RNA. Okay. The next question, the codon for anticodon, that is 3 prime UUA 5 prime. So it is very clear. So anticodon we should identify. So but here they have given already anticodon only, then you should identify the codon. 3 to opposite to the 3 prime, then it will be 5 prime. UA means AAU, and there in anticodon 5 prime is there, in codon 3 prime should be there. Okay, it's very easy actually, but opposite prime with respect to the anticodon to codons or codon to anticodons, like that they will ask the questions in exams also. Next question, the cytosine base inserted in the beginning of DNA codes, see write down ATG, ATG, ATG. Okay, so here will produce, they're asking cytosine base inserted in the beginning of DNA codes ATG, ATG, ATG. Okay, so then whenever we see this one, what are the things it will produces? So you just see that whatever they have given here at the same time. Okay, so what we should identify, that is nothing but observe the table which is given the codons, right? If you see that one, then you can identify, that is CAT, GAT, GAT and G. Okay, are you getting? Fine. Yeah. The next question, the regulation of gene expression, it is commonly known as Lactose concept. Okay, in prokaryotes which level of gene expression is absent? They are asking absent, not present. In prokaryotes which level of gene expression is absent? So it is actually a direct question. Processing level, the processing level of gene expression is absent. Okay, because the transcription level is present. Even transport of messenger RNA from nucleus to cytoplasm, it is a present. Translation level is also present, but processing level is absent in case of the prokaryotes. Okay. The next question, it is also related to the same topic. Operon, what is operon? I told no, it is a direct definition, a set of closely linked genes regulating a metabolic pathway in prokaryotes is called operon. In general, we can also say a set of closely linked genes, okay, which is responsible for the regulation of metabolic pathway in prokaryotes. Okay, I told you clearly while we are discussing about the Lactose concept. The next question, it is also related to the regulation of gene expression. Regulatory genes are located. See here the question is directly where it is located, isn't it? In front of structural genes. See the structure of the unit. Okay, so then you can identify the regulatory genes are located in front of structural genes. Okay. The next question, it is also related to the same topic. 3 enzymes required to break down lactose into glucose and galactose. So just see the Lactose concept so that you can easily identify. There are 3 names of the enzymes, okay, which is related to the conversion of lactose into glucose and galactose. That is nothing but beta galactosidase enzyme. Another one is beta galactosidase permease, and the third one is beta galactosidase transacetylase. Okay, in short forms we can write beta galactosidase, permease and transacetylase. Okay, these 3 name of the enzymes are related to the breakdown of lactose into glucose and galactose. Okay. The next question, this is related to the HGP, that is a human genome project, which is not correct statement about the human genome. So here we should identify not correct. See, whatever they have given these statements which is related to the human genome, but we should identify incorrect or not correct statement, remaining and all correct. Let me to read one by one. The first option, largest human, that is a non-human gene, dystrophin on X chromosome, contains 2.4 million bases. Yes, it's correct. Okay, then 99.9% nucleotide bases are identical in all the people. Yes, that is also correct. Then about 1.4 million locations, the where single base DNA differences SNPs occurs in the humans. This is also correct. Option D, Y chromosome has most genes and chromosome first has the fewest. False. It is incorrect statement because they have given ulta, that is a vice versa. Actually X chromosome has the most genes, isn't it? That is a 2968, whereas that is the chromosome 1 has the fewest, that is a Y. Actually Y chromosome has the fewest, that is a 231, but here they have given the ulta or the vice versa. So that's why option D is the correct option with respect to the wrong or the incorrect statement about the HGP. Okay, so this is also related to the HGP. Human Genome Project was closely associated with bioinformatics. I told no, so it is related to the bioinformatics. By using this technology, they will store the data, okay, in the supercomputers. Okay, so there is nothing but Human Genome Project is also known as mega project, isn't it? So that's why generally it is directly associated with bioinformatics. The next question, the basis of DNA fingerprinting. I told no, it is also known as DNA profiling. Okay, direct one, the polymorphism in sequences. Okay, polymorphism in sequences. That's why each and every individual present in this world, they're all different with respect to one another. Okay, poly means many, morphism, morphological expression characters with respect to the sequences. The next question, the DNA fingerprinting technology, a size of, okay, that's a size of VNTR, variable number tendon repeats, varies from, okay, so I told you it is related to the 0.1 to 20 kb in theory only. I told you clearly, and I am actually, while I am telling about the DNA profiling or the fingerprinting, I told you clearly this question can be asked in competitive also. Okay, so that means the size of variable number tandem repeats varies from 0.1 to 20 kb with respect to the different organisms. Okay, the last question, okay, which one is not involved in DNA fingerprinting? Okay, see whatever they have given here, the options, except one option, all are involved in the DNA fingerprinting technology. The digestion of DNA by restriction endonucleases, it is involved. Gel electrophoresis, it is also involved. Blotting to synthetic membranes, that is southern blotting technique, it is involved. Reverse transcription of hybridized DNA, it is not involved. Okay, so auto radiography, it is also involved. So that's why the reverse transcription we were not discussed in case of the DNA fingerprinting technology, isn't it? So that's why option D is the right option with respect to the not involved, okay, in the DNA fingerprinting technology. Okay, so let it be. So these are all about the competitive, that is a NEET or CET part with respect to the 6th chapter. So in the next coming classes we will start another important chapter, evolution. So before we are going to the the next chapter, please recall, repeat complete chapter. If you even if you are having any the doubts, means please let me to clarify. Okay, you have to ask. Okay, then only we can tell you and we can clarify all those things. Okay, so then only, so further chapters or the topics will continue. Okay, so anyway, thank you, have a nice day.